Chapter 18, Part 2, DNA Damage Repair Systems, or if you like, DNA Repair Mechanisms. Our cells have evolved mechanisms to repair DNA damage, so when it's broken, we have the ability to fix it. A number of pathways repair changes in DNA, including the mismatch repair, or MR pathway, the direct repair, or DR pathway, the base excision repair, BER pathway, nucleotide excision repair, or NER for short, non-homologous end joining, NHEJ for short, and last, homologous recombination, or HR for short. So those are the 6 pathways, mechanisms for repairing DNA damage. And here's a snapshot of 5 of the 6, because one of the 6, direct repair, does not happen in eukaryotes. That only happens, well, it doesn't happen in vertebrates anyway. It happens in E. coli and yeast single-cell organisms, mostly prokaryotes. But we've got different types of damage. We've got single-stranded breaks, double-stranded breaks, bulky adducts that distort the DNA, base mismatches, insertions, and deletions, which we talked about. And you can see that there's all sorts of different ways to repair these. So the base excision repair repairs single-stranded DNA. Double-stranded breaks can be repaired by either homologous recombination or non-homologous end joining. Bulky adducts that distort the DNA, such as UV radiation, can be repaired by nucleotide excision repair. And then base mismatches, insertions, and deletions can be repaired by the mismatch repair pathway. So looking at single-stranded break, and also mismatches, but this affects one of the 2 DNA strands, these that we've got coming to a town near you. We've got human hereditary colon cancer that can be caused by a defect in this repair system. So you can get the damage, and your repair system, a mismatch repair system, is supposed to fix the damage. However, sometimes you can have a mutation in your system for repairing, and that can lead to cancer, because you're going to get accumulation of mutations. So this method involves methylation in prokaryotes, and for sure, and it is important for correcting mispaired bases. So mismatch repair, colon cancer, methylation. All right, so in prokaryotes, where we've done more of the studying in that model organism, mismatch repair does the following. It replaces a nucleotide on the new strand of the mispaired bases. So we've got here in this diagram, we've got an old DNA template shown in green, and kind of a reddish-orange color, we've got our new DNA. And you can see we've got a mismatch right here, and so there's a little bulge in the DNA. So G, it does not complementary base pair to T. Also, on this piece of DNA, we've got a little region with the sequence GATC on the new strand and CTAG on the old strand, and attached to the old strand is a methyl group, but there is not a methyl group attached to the new strand. So there's my little color code key. New strand, kind of orangish red, old strand is green. So, you know, you make this mistake, right? And how does the cell know which one is the mistake and needs to be corrected, and which one is the one that is the original? Well, it's that little methyl group that helps out with that. So there are a complex of proteins that are going to recognize this methyl group on the old DNA strand, and they're going to recognize this mismatch G from T in this case, and there's going to be a coordination of efforts to fix that mismatch. So first of all, we've got MUTH is a protein that is going to recognize the methyl group. It's going to recognize the signal CTAG that has the methyl group on it. So this is the protein that recognizes which one is the old strand and which one is the new strand that needs to be repaired. MUTS is going to find the mismatch. Mismatch has an S in it. MUTS has an S in it. Methyl has an H in it, and MUTH has an H in it. So hopefully that can help you remember what protein is what. Yes, you do need to know which protein is doing what. MUTL, all right, is linking the two so that we get a little communication between the two. So they're like, okay, here's the methyl group. This is the old strand. This is the new strand. And this guy's going, okay, here's the mismatch. We need to get rid of this, according to you, the G, not the T. And so they do that. They have an exonuclease that comes in here. So we get protein identifies the GATC of the old strand. Get two proteins that recognize the mismatch. Exonuclease is going to come in and remove the DNA strand from the methyl group at the GATC to just beyond the mismatch, as you can see from this diagram. And then what happens? We get a synthesis of a resynthesis of the DNA. This time an A was put across from the T. Thank goodness we corrected that mismatch. Who made the new DNA? Well, DNA polymerase, my friend, and DNA ligase finished the repair job. These are enzymes that you've already learned about from DNA replication. So sun is gorgeous. We like sunny days, but the sun also causes UV damage to our DNA. So DNA repair mechanisms that fix UV-caused DNA damage do exist. And here's what typically happens with UV. So we have UV-induced mutation. A pyrimidine dimer is shown here. What does that look like? Well, usually pyrimidines, which you know are the nitrogenous base part of the nucleotides in this DNA backbone, they're attached to the DNA backbone, sugar phosphate backbone. Usually they are not connected to each other. They are separate. But when UV light comes along, this is what can happen. We can get covalent bonds forming between these pyrimidines, and now this is called a pyrimidine dimer. And we are showing you the thymine-thymine dimer, but you can also have a cytosine-cytosine dimer or a cytosine-thymine dimer. Why is it that purines are susceptible to this? Don't really know the answer to that question, but we do observe that that's what tends to happen when you get UV irradiation. You get pyrimidine dimers. Okay, so it makes your DNA bulge a little bit. That's not ideal for DNA replication, passing the genes down. So we have some repair mechanisms to deal with that UV-caused damage. One is direct DNA repair, bacteria and yeast, and two is nucleotide excision repair, aka NER. And this is what we find in vertebrate organisms. You can also find it in prokaryotes. So direct DNA repair is shown here. So I've got some lovely E. coli on the beach, and believe you me, they're on the beach too when you are, and they're in the water and they're just enjoying the sun. But the thing is that they get some damage. So they get some UV damage. And what they have to repair that UV damage is a photolyase repair enzyme. They don't need to replace nucleotides. They don't need any exonuclease. None of that fancy stuff we just saw with the mismatch repair. Oh no, they just have an enzyme that only fixes damage directly, and it uses that light that caused the damage, the light energy, to break that covalent bond linking those two pyrimidines. So this is a little diagram of that. You get, here's your pyrimidines, TT. The dimer is symbolized by this little triangle in between here. We get a thymidine dimer, and then we have photolyase come along shown in green, but I decided to also do it in blue. Photolyase is going to use visible light to break, just physically break that covalent bond between those thymine dimers, and then it leaves. So who has photolyase? Budding yeasts, Saccharomyces cerevisiae, bacteria such as Escherichia coli, but not us humans. More about how to fix DNA. We got nucleotide excision repair, or NER. This works in eukaryotes. It can also work in prokaryotes. It's much more complicated in eukaryotes than in prokaryotes, but both systems have it. When proteins responsible for DNA repair mechanisms are mutated, you can get human disease because you can't correct those mutations. Our cells have evolved mechanisms to repair DNA damage. When these repair mechanisms don't work, mutations are going to accumulate, resulting in diseases such as cancer. Please remember the diseases in yellow shown on this table. We've got xeroderma pigmentosum, hereditary non-polyposis colon cancer, and Werner's syndrome. The symptoms and the genetic defect. So we've got different genetic systems that are broken. And xeroderma pigmentosum is the nucleotide excision repair system that's broken. In hereditary non-polyposis colon cancer, which we just looked at earlier, it's a defect in mismatch repair. In Werner's syndrome, we get premature aging and cancer. This is a defect in homologous recombination. So here's xeroderma pigmentosum, or XP for short. Thank goodness we've got a for short, because that's quite the words. Genetics: defect in nucleotide excision repair. It's an autosomal recessive disease, multiple gene involvement. And you can see from these patients over here that they have extreme sensitivity to sunlight, abnormal skin pigmentation, and early onset of skin cancer at a rate that is 1,000 to 2,000 times more frequent than unaffected individuals. So these kids literally cannot go out in the daytime. In fact, they have summer camps for them that occur, all the events occur at nighttime. They are that sensitive to the sunlight. Cause of XP: mutation in genes coding for proteins in the nucleotide excision repair pathway or system. UV damage creates pyrimidine dimers. Broken NER system cannot repair them. So here's how an NER is supposed to work. You get damage to the DNA. It distorts the configuration of the DNA molecule, shown here in red. An enzyme complex recognizes the distortion resulting from that damage. So this big slab here is the proteins, the enzyme complex. The DNA is separated, and single-strand binding proteins stabilize the single strands. So this is very similar to replication. We have to unwind the DNA. We've got some single strands of DNA exposed and single-stranded binding protein to stabilize those while we do the repair. So it kind of reminds me of taking a jack to your car to lift up the car so that you can get to the wheel and then change it. Here we're like cranking apart the DNA with this enzyme complex, and we're holding it up not with a jack but with single-stranded binding protein so that we can now repair this region. An enzyme cleaves the strands on both sides of the damaged region, boom and boom. Part of the damaged strand is removed, and then the gap is filled in by our friend DNA polymerase and the other friend, sealed by DNA ligase. So just like in mismatch repair, we had to have DNA polymerase and DNA ligase come in. They have their functions in DNA replication and also in DNA damage repair. All right, so we get our new DNA. This is just a little fun animation of what I just said to you. We got UV damage and nucleotide excision repair. We get proteins that recognize that damage. We unwind the damaged area. we do a cut on the 3' end, 3' incision. The protein here is called XPG. XP for xeroderma pigmentosum, letter G. That means there was an A, B, C, D, E, and F. And in fact, XPF is what's going to give you a cut or incision upstream of the damage, 5' of that damage. Then you're going to have removal of the damaged DNA, and then resynthesis of that damaged DNA using the other piece of DNA as a template. So basically, this is why XP patients are sensitive to sunlight. Usually you have healthy DNA. When UV hits it, you get damaged DNA, and the nucleotide excision repair system fixes that damage, and you have a healthy DNA. That doesn't work in, that system doesn't work in these XP patients until you have no repair. So another repair mechanism or system or pathway is base excision repair, BER. Here's a mutation called depurination. So depurination. So that means we got a purine right here, and we basically don't have it anymore. We depurinated it, and what we have left is called an apurinic or AP site. Apurinic site is what is created right here when we get rid of that damaged nitrogenous base. Well, how did it get damaged? You might ask. Well, here are some ways that it can get damaged, some base damaging agents. You could have reactive oxygen species that have extra electrons that can damage that region of your DNA, and then it needs to be repaired. You can have oxidizing agents. Again, similar sort of thing. We're going to damage DNA, just like you're going to get rust on your steel parts of your bicycle, for example. Oxidizing agents are causing that. Oxidizing agents can also cause problems with your DNA molecule. They can damage them in a similar manner. UV radiation, of course, which we've already talked about. So we could get a bit damaged base, and instead of being able to break the covalent bond apart, we just remove the base. So that's the first step when we do base excision repair. We're excising or removing the base. We get a damaged base. You can see it right here. It's looking a little funky. And then the thing that removes it is an enzyme called DNA glycosylase. And it generates what we call an AP site, an apurinic site. A meaning it's gone, it's lost, apurinic. Then AP nuclease is going to recognize, it's going to say, hey, you're missing something there. You're missing your base, so it's going to take out the rest of that nucleotide. And then DNA polymerase is going to come in. And it's going to repair. DNA polymerase adds new nucleotides to the exposed 3' hydroxyl group. This is the grand finale. And then DNA ligase. Hey, it's the dynamic duo again. DNA polymerase and DNA ligase. Where did we see those before? Oh yeah, we saw that in mismatch repair, and we saw that in nucleotide excision repair, and here it is again in base excision repair. Are you seeing a pattern? I am. The only thing we didn't see it in so far is direct DNA repair. So a small note on base excision repair. The DNA polymerase that comes in, there are many DNA polymerases, as you may be aware of. In eukaryotes, it's DNA polymerase beta is used in the base excision repair pathway, and it can insert the wrong nucleotide. So you can have a result of 20,000 to 40,000 mistakes that are corrected per day but some may not get corrected in your genome has an average of 10 errors per day left by BER. So it's fixing lots of them, thousands of them, but it might leave a few for you. And those, where those mutations are, They could do absolutely nothing to change your phenotype, or they, could do something drastic. It just depends on location, location, location. So now we're moving into double-strand breaks. Double-strand break repair systems. dsDNA is double-stranded DNA. So, some of you may know your history. Double-stranded DNA breaks can result from gamma radiation exposure. So over here is a plane and a man standing in front of it, and the name on the plane is the Enola Gay. If you know your history, you'll recognize the name of that plane. It was the plane that dropped the atomic bomb on Hiroshima and killed thousands of people in an instant, and was part of what brought World War II to an end. This happened in 1945. The A-bomb dropped and killed 90 to 140,000 people. It's hard to know because it was hard to find pieces of people. 3 days later, the U.S. hit Japan again at Nagasaki, killing 60 to 80,000 people. I don't know how we are at peace with Japan now, but we are. But man, we did some heavy damage to them. And yeah, World War II, damage was done to us. Fortunately, we were protected from most of it because of the Atlantic Ocean and the Pacific Ocean. Nowadays, we've got a different type of warfare, and it's happening on the thing that you carry around with you all the time, your cell phone. People can watch what you're doing. People can advertise to you based on what you do your Google searches on. If you guys do Facebook, I think your generation doesn't really do it, but you do other social media things, and you can be targeted for your ads, and it's a different and more clever type of warfare than bombing, but nonetheless, it is happening. Dr. Key will move back to science now. Many more exposed versus control group. They looked at these folks that survived Hiroshima. They were further away from the blast. They did see somatic mutations increase. They saw radiation sickness and cancer rates increase. They looked for germline mutations, and they did see increases in birth defects, chromosome abnormalities, and gene mutations in children born to those exposed to this gamma radiation from the atomic bomb. If this young man here doesn't need to have an atomic bomb drop on him to end his life early, because he unfortunately suffers from what is called Bloom syndrome, and he's got a defect in a gene product called the Bloom's helicase, or BLM1, and that helicase is very important if you get damage that you would sustain from a blast such as this, where you get double strand breaks. So this type of damage that they're talking about over here at the germline and the somatic mutations, a lot of them are double-stranded breaks, especially when you're talking radiation sickness. That means you got such a high exposure that your repair systems just could not keep up with the damage. But this young man, just being alive can kill him because he can't repair double-stranded breaks at all because of a defect in the Bloom's helicase gene that makes his protein defective. Because patients with Bloom syndrome cannot repair double-stranded DNA or dsDNA breaks, they have the following symptoms: photosensitivity, male sterility, because every time you try to do meiosis, you've got that crossing over happening during prophase. Well, he can't uncross the crossover, so sterility. Increased rate of all types of cancer, 50% chance of occurrence. Mean life expectancy is just 24 years old. That's how much we have to repair mutations on a daily basis. So 2 ways to fix double-stranded DNA or dsDNA breaks. One is called non-homologous end joining, NHEJ for short. And the second one is homologous recombination. Yes, that's the same thing we were talking about with crossing over and creating meiotic genetic diversity through meiosis. You need homologous recombination to repair damaged DNA as well. So there's 2 ways to repair double-stranded DNA breaks. What we're seeing here is one double-stranded piece of DNA, and here's the second double-stranded pair of DNA. We get X-rays, gamma rays. They hit them, boom, you get a double-stranded break. You can either get, so non-homologous end joining involves some proteins that recognize that break. You get X-ray repair, cross-complementary protein 4, or XRCC4 for short, and other proteins. So one of them is this XRCC4 protein that's going to recognize this damage and fix it. And how does it fix it? It just slaps the 2 ends together. So what's ever missing in the middle is gone. So yeah, it's like duct tape. And it's error prone. You're going to have deletions. You could have insertions, and you can have those things called translocations, which are when non-homologous chromosomes cross. And somebody needs to ask me that during your virtual time. And I might be able to draw it at the end of this if I have any time. So it may be responsible for translocations that lead to cancer, for example. The other type of repair mechanism is homologous recombination, and it's the better one, but it takes time. So you have to get your double-stranded break. Now this is going to go quick. I'll back up when I may have to back up a little bit. So double-stranded break in one DNA molecule. What are you going to do? There's nothing to repair it with. Okay, so you're going to, okay, that goes fast. You're going to, you do, the first thing is resection by exo or endonucleases. Then you get, oh dear, let me go this way. You get a single-stranded DNA created, and this blue one invades the orange one, strand invasion. The bacterial protein that helps lead this strand to the other double-stranded DNA is called a Rec-A. So here's one homologous chromosome. Here's the second homologous chromosome. So you're having to go over to your homologous partner to get the DNA fixed. You get to this point of repairing the DNA synthesis, and now you have to have a helicase to get that strand back over and finish repairing the broken homolog of DNA, the broken chromosome. So the complementary sequences anneal to each other, and along comes Bloom helicase, and it's responsible for unwinding this crossover event, this homologous recombination. As long as this protein is working, you're going to be able to fix this double-stranded DNA break. But if that protein is not working, as it is not in patients with Bloom syndrome, then they cannot unwind this DNA, and so they cannot complete the repair. Okay, but in a normal patient, you would be able to unwind it and complete the repair. So here's this table of 1, 2, 3, 4, 5, 6, all of the 6 mechanisms that we've talked about. Summary of common DNA repair mechanisms and pathways in table 18.5. So please study the repair systems and the type of damage that results from, the type of damage that they typically repair, shown in the second column. Summary, so we talked about 6 DNA repair pathways to repair DNA damage. And that is all.